Hi, sorry, I'm back. I have no idea what just happened. It just like conked out on me. But anyway, I'm glad you guys are here. So obviously you get to watch these now for free. But just a heads up, so Fiveable Plus is the additional program that you guys can sign up for. You get access to replays. Otherwise, this is your only opportunity to watch. You can live stream, or we'll have live stream notes, cram sessions for subjects, lots of good stuff. So it's definitely worth being a member. Membership is just $20 before April 1st. After April 1st, for all those kind of late to the game reviewers, it'll be $35. So you save 55% if you sign up early. And they're good until July 1st. And then they would need to be renewed for the 2019-2020 school year. But it would get you through your AP season, and you would have access to all of our AP subjects. So definitely worth it if you plan on tuning in, want to see replays, all that jazz. Okay, so as I was saying, we're talking non-Mendelian genetics and pedigrees this afternoon or this evening. I'm going to show you guys put my PowerPoint slide up. Okay, and we'll go. I'll check back periodically because I'm using regular PowerPoint now because it's easier for me to write as opposed to just Google Slides. It's a little harder for me to check back on you guys and make sure that you're doing okay. But I promise I'll check back periodically. Okay, so non-Mendelian genetics and pedigrees. So first and foremost, what in the world is the purpose here? I try to start with that always. And the bottom line is that whenever you've talked about genetics in whatever class that may have been, you've probably talked about dominant and recessive traits and Mendelian genetics, whether you knew they were called Mendelian genetics or not. The problem is the vast majority of traits are not inherited this way. So we need to learn a little bit more about a few other inheritance patterns in order to kind of understand the inheritance of some other traits and genes. So everything that we've looked at so far has had only two alleles, a dominant and a recessive. When we were doing chi-squared, dihybrid crosses, monohybrid crosses, and talking about Mendelian genetics, all of those obviously just involved two alleles and a single gene that we were looking at with very predictable inheritance patterns. There's so much more. And again, we're not even going to cover half of it because some of it is just way above and beyond the scope of what you would need to know. But there are a few other inheritance patterns that you should be aware of. And at the end, I have a really great way for you to remember what pedigrees would look like with different inheritance patterns. And that's a really big one for the AP exam. They like to throw pedigrees on there, and you have to be able to analyze them and determine the inheritance pattern from them. So that'll be coming up at the end of this session. Okay, so the major non-Mendelian inheritance patterns that we're going to talk about tonight are codominance, incomplete dominance, multiple alleles, and then sex-linked traits. None of these are all that hard at all, which is the good news. You just need to be familiar with them and know that there's another way that traits can be passed on from generation to generation that might look differently. So we'll start by talking about codominance. The best way to remember this is if you can remember that co means together or both, like a co-captain. If you're the co-captain of a team, then you and your other co-captain are equally captains together. You share that responsibility, cooperation, working together towards an operation of some sort, all sorts of words, but co again means together or both. So if alleles are codominant, if they show a codominant inheritance pattern, then what that means is both alleles are dominant. There's not one that's more dominant than the other. So we don't have that dominant and recessiveness. We just kind of have a dominant, and then some blend of those two. The way to think about dominance, it really depends on the heterozygote. Okay, so when we look at the heterozygote, if alleles are codominant, then we should see both alleles present in that heterozygote. And so we'll see this below. So feather color in some breeds or species of chickens is codominant. So if you have a white feathered chicken crossed with a black feathered chicken, you will get this checkered chicken that you see in the bottom right hand corner. And so what you can see there is there's both black and white feathers being shown. Okay, so the heterozygote shows both colors, which is again that codominantness. Right? So very, very simple. The only thing that's changing is the heterozygote. So instead of now one being more dominant and that's what shows up in the blend, they both show up in the blend. So if two checkered chickens are crossed, what is the expected outcome of the babies? All right, so I'm going to draw a little Punnett square here. So let's say that we have big B for black, big W for white. Oop, that's not what we're crossing. I'm saying, wow, I'm losing my mind. Sorry, everyone. Do I have an eraser on this thing? I'm learning. Ooh. Yikes. Okay, I'll just draw a new one because, oh, there it is. Yes. Perfect. They make that very easy. All right. So BW, because we have two checkered chickens. I wasn't reading my problem carefully. And so then we have, when we cross these guys, wow, I really need to get a mouse. We have one offspring that's going to be black, two offsprings that are going to be checkered, and one offspring that would be white. So we would have 1 out of 4, or 25%, which would end up being 25 eggs, right? If they have 100, would be black. We have 2 out of 4, or 50%, which would be 50 eggs. That would be checkered. Okay, and this is the big difference. Again, this is the only thing you kind of have to keep an eye out for. Hopefully you're like, wow, this is really, really easy. Because it is, but you just have to be aware that these heterozygotes are now going to not look black or white, but their own phenotype. Okay, so this reflects back to, if you were with us last week when we talked about chi-square, the possible phenotypes. Now we have 3. You can be black, white, or checkered, not just black or white. And then the last quarter, or 25 eggs, would be white then. Okay, easy, truly easy enough. But you just need to know that it's possible that things would be inherited this way as opposed to in the normal way. Okay. All right, another quick example with this, and then we'll move on. Short-horn cattle, color is codominant for the short-horn cattle. So a red cow is genotype big R, white cow genotype WW. Heterozygous cattle are called Roan, and they're spotted red and white. Maybe you've seen something like that before. When we cross these guys, we're going to see any number of things from them, okay, with those heterozygotes, again, being their own specific phenotype, okay? So, oops, I forgot to paste the problem in here. Woof. Okay, so let's just say we have a red bull, okay, crossed with a roan cow, okay? So red bull, big R, roan cow, big R, big W. What percent are we going to expect of all the different phenotypes from this cross? Well, our top 2 boxes are going to be red, like the bull. Boxes are going to be roan, like the cow. Okay. So we should have 50% of our offspring being red, and then 50% of our offspring being roan for that red and white spot. So 50% for each of those, easy enough. Okay. Again, pretty basic. Just need to know it exists. Only thing it affects is the heterozygote. Okay, I'm gonna real quick check back on you guys, make sure everyone's doing okay, no problems. Looks like we are. Perfect. We'll keep moving on then. This is an easy week, guys. It's just taking a little break after chi-square last week, the big one. Okay, so incomplete dominance then is going to be a different inheritance pattern. So we have codominance, both alleles are dominant. Incomplete dominance, neither allele is dominant. Okay, so what happens here, and again, remember this really just depends on the heterozygote. Instead of being red or white, we have some sort of blend. In this case, we have pink for these flowers. So neither red or white is showing at all. It's not like red and white spots, which would be an example of codominance. Instead, we have just this blend. Neither one gets to beat out the other. They just kind of meld into this middle ground. Okay, so just another option. If you don't like the fact, some of my students don't like the fact that in this particular example from the textbook, white is given 2 small r's because it makes them feel like, oh, then there is a dominant and recessive. Then you can certainly make this 2 capital W's instead, and then the heterozygote would just be RW. Totally fine. The nomenclature itself doesn't really matter as long as you understand the concept and what happens to the heterozygote. Okay, So for incomplete dominance, what would happen in the following situations? If we have a red flower and a white flower, okay, so red and white. Easy enough. You should be able to tell hopefully already, and that's the goal here, right? You shouldn't have to really draw out a Punnett square each time, that every single one of the offspring is going to be heterozygous, and therefore we should have 100% pink. Okay? This is a terrible thing to write with, but you know, gotta do what you gotta do. All right, if we have pink mixed with white, okay, now we have big R, little r, we have that heterozygote mixed with our white flower. We're gonna get half of our offspring being pink and half being white. Okay, so 50% pink. Oh gosh, disaster. Here we go. 50% white. Okay. And then the last one, which you've probably already worked ahead while I'm struggling through writing this, is a pink flower crossed with a red flower. So pink, heterozygote, crossed with red. And again, you can change the letters if you like that better. No worries there. You should get, in this case, 50% red and 50% pink. Okay, so again, easy enough. It's just that this time, so we have 50% red. I'm just going to write R, save myself, and U, and 50% pink. Okay, only difference is that heterozygote. Not too complicated, easy enough. Okay. All right, so incomplete dominance and codominance. And again, by thinking about the names long enough, you'll be able to figure out which one is which if they tell you that something is incompletely or codominant. All right, a little bit more confusing is going to be something involving multiple alleles. So again, as I've said, and even in the last two examples, we only had two alleles, right? You might not have had a dominant and a recessive allele. In fact, we had two dominants or two non-dominant alleles in our past two examples, but there's only been two, okay? But that doesn't happen all the time either. So many genes actually exist in multiple forms, so they actually have multiple alleles besides just those two options. So what we do here is we actually write the allele in the upper right-hand corner like a superscript. And you can see that here, and then you'll be able to see it on the next page as well. But we mark it that way because we can't just have two letters anymore, just because we have more than two options here. So the two classic examples of multiple alleles, we have a certain breed of bunny, their fur color. If you've ever seen chinchilla bunnies or Himalayan bunnies or albino bunnies, their fur color is controlled by 4 different alleles. Okay, and then we also have human blood type, which I think is incredibly interesting, and that's what we're going to focus on tonight because it's more relatable. Okay, so with human blood type, there's 8 different types. We're just going to focus on the 4 letter types, if you will, so O, A, B, and AB, and not worry about the difference in pluses and minuses. But if you're unfamiliar with blood type, you can actually take a look at this picture. It's pretty awesome. So the most common blood type is O positive. The rarest blood type is AB negative. And another second most common is A positive. That's my blood type. But what you can see is each of these demarcations. So if you have blood type A, you'll see here you have, in this example, purple spikes coming off of your blood cells. But you have a certain protein that's on the surface of your blood cell that we have named blood type A. Okay, so you can see in all 4 of these examples that contain some part of part A blood, either A minus, A plus, AB minus, AB positive. They have these projections off of them that the body reads as this certain blood type. Okay, and this is going to come in handy. So then you have different proteins on your cell's surface if you have B blood type or AB blood type. And then you have, yet again, a different demarcation if you are O, A, B, or AB positive, and no demarcation if you are O, A, B, AB negative. So you'll see that O negative actually has no projections, no proteins off of the surface of the cell. Okay, so what happens is, so I'm A positive. Okay, so if I needed a blood transfusion and they gave me B positive blood on accident, my body all of your white blood cells, and we're going to talk about the immune system in a few weeks, they scan your cells to determine what is you and what is not you, right? You have different complexes, different proteins on the surface of your cells that your body recognizes as you. So if all of a sudden these cells enter my body is going to scan those blood cells and realize, hey, this isn't you. We need to fight this, right? And you can get really, really sick. Your body's going to kill all of those blood cells. You can go into shock, and it can be incredibly damaging. So that's what makes blood typing so important. Now, the unique thing is that me being A positive, I can get blood from A positive individuals, but I can also get blood from any of these three. I can get blood from O positive because my body will recognize the positive projections. It's not going to notice that there's not a protein, a blood type protein sticking off the surface, right? I can get blood from A negative because, again, it's not going to notice the lack of positive protein. It's just going to notice the A protein and be like, hey, cool, this is us. Or O negative. We call O negative the universal donor because they have no surface projections. They can donate to any blood type, including themselves, but they can only receive blood from other O negative individuals because their body would freak out and attack any other blood cell because it has some sort of projection or protein on the surface. On the flip side, AB positive, which is a very rare blood type, is considered the universal receiver because they have all three proteins on the surface of the cell, A protein, B protein, and the positive protein. So any other donor can donate to them and they will be just fine because their body recognizes all proteins. Okay, so that's just kind of an interesting, cool thing about blood type. You don't have to memorize any of that for the AP exam, but it's really cool to know about. So the other interesting thing to know about blood type is that it's both codominant and dominant recessive in nature. I'm going to write this on the next slide, but O is actually recessive. So this allele here is recessive. So you can see in this example, if you have an A-type allele and an O allele, then you have blood type A, right? And that should kind of make sense because we don't have like an A-O blood type. So B and the O allele is just going to give you type B blood. And the only way that you would have type O blood is if you got those two recessive O alleles. Okay, and we're going to talk about this a lot more on the next slide. So here are the phenotypes and genotypes of our four major blood types. Okay, so things to notice are that blood types A and B, there are two different genotypes that give those phenotypes, specifically AA, AO, BB, and BO. Okay. AB, you have to have an A and a B allele. And then for blood type O, you have to have two O alleles. So the way that we can kind of show this and keep it straight in our mind is by having capital I's for our A and B alleles, and then lowercase I indicating that they are recessive. So the important thing to remember here is that A and, this is so hard, A and B, there we go, are I'm just going to have to get really good at cursive. It's bringing me back to a dark place. Codominant. Okay. Getting better. Codominant. And O is recessive to both. Okay, so O is recessive. Those are not cursive S's. They're very loopy. Okay. Cool. So knowing this stuff, we're going to just start applying. We're going to do a couple practice problems here. So what is the expected genotypic ratio among children born to a mother having the genotype AO and a father with the phenotype AB? Notice that they just say phenotype, but for AB, we know the genotype because it has to have an A and a B allele. Okay, so if we have AO crossed with AB, and maybe you're thinking back to like what your blood type might be. I didn't find out my blood type until college when I donated blood for the first time. But I was then able to figure out my genotype because I have A positive blood, but my mom is blood type O. So I know that this is my genotype right up here, AO, because she has to be OO in order to be blood type O. Anyway, all right, so once we cross these, it's the same as ever before. We're just carrying an extra letter this time. Okay, so we have one that's going to be blood type A. We have another that's going to be blood type A, because remember, this is recessive. Okay, We have one that's going to be blood type AB. And our final one is going to be blood type B because we have the B allele and then we have that recessive O allele. Okay, so the genotypic ratio, it would be 25% A, or sorry, 50% A, 25% AB, 25% B, 0% O from this cross. Okay, and that one was pretty easy because they gave us all the information that we needed. All right, so I'm going to just check back and make sure everybody's doing okay. Oh, shoot. Oh my gosh, I'm so sorry, guys. Why is it frozen? No. Oh no. Have you guys just been listening to me this whole time while I'm absolutely... Oh no. Does it work now, guys? Could you see the correct slide? Could you see the blood cells? This is so weird. It worked last week. Oh dear. Eee. Shoot. All right, let's try this again. I can't believe you guys just listened to me that whole time. I am so sorry. Let's try this again. Where are you? There we go. Okay, let's see now. I'm gonna go into this. Did you guys see the alleles then? Ooh, gosh, why do you keep disappearing? Or did you just see? Oh my gosh, why is it doing this? Shoot. Was it following along? Yes or no? Anyone? When I was presenting, when it went to full screen, was it following along? Okay. Okay, multiple alleles is what I was on. So I'll pull that back up. Just make sure that this is following along now. Here's what you missed. We talked about codominance. Okay, so there's your checkered chicken that you didn't get to see the first time. We talked about incomplete dominance. So there's your red, white, and pink flowers, as well as the crosses for them. Multiple alleles is what we just talked about. So here you can see the different blood types that I was telling you about with all the cool surface markers. Okay, so you have all your negatives on the top because they're lacking this green in this picture. Positive protein, so you can see what the A protein, the B protein, and the AB proteins would look like on a blood cell. And then once you add the positives, obviously, then you have multiple projections from each cell representing those blood types. Okay, I'm going to exit out again just to make sure that you're all with me and that worked. Did it go through the slides when I was going through them with you? You could see the incomplete and codominance. It stayed on one? What the heck? Oh, goodness gracious. Okay. Maybe I just have to do this and see if it'll switch out. No, it's still not going to switch. Oh my goodness. What's going on? All right. Stay tuned really quick. I'm going to try to upload this to Google and see if it'll let me present. I don't know what's going on. Hopefully this wasn't happening all of last week. If it did, no one told me. Okay. All right, I think I converted it to Google Slides. It's not going to let me write on it like I was, unfortunately. Shoot. What the heck happened there? All right, here we go. Okay, ooh, we'll try this. All right. I don't know why everything's taking together forever. Maybe my internet is crapping out or something. This is strange. We'll zoom ahead. All will be well. Okay. So thank you again for following along even without being able to look at the pictures. So here is what we were talking about. This is going to be the most important visual with the different blood typing and the different proteins on the surfaces of the cell. And again, here you can take a look at the different inheritance patterns for these, but those are highlighted on the next page even better. So we have obviously only 4 phenotypes for blood type. We have 1, 2, 3, 4, 5, 6 different genotypes possible. Okay, and this is just for being A, B, or O, disregarding to the positive and the negative, which adds a whole other factor, and it's controlled by a different gene. So if you have blood type A, you can either be AA or AO. We show that with that lowercase I, like I was saying. If you're blood type B, you could be BB or BO with the lowercase I again. AB, you're just going to be AB. And then O is 2 O's because that's our recessive trait. Okay, so this is what I was just showing on that other PowerPoint slide that unfortunately you guys couldn't see. But if you have AO crossed with AB, from that Punnett square cross, you would have, let's see, oh, shoot. I can't write on these, and I can write on my PowerPoint slides. So let me you might just have to skip this section, and I'll get back to it next week. But you would have, obviously, the mom giving either an A or an O. So you could have an A with an A from the father for blood type A, an O from mom, and an A from the father for AO, an A from mom and a B from the father, which would be AB, or an O from mom and a B from the father, which would be BO. So 50% A, 25% AB, and then 25% O there. Okay, so we can just kind of think about these problems since I can't write them out for you. One parent has blood type A and the other has blood type B. What are the genotypes of the parents if they produce children with only blood type AB? Okay, so there's going to be 3 different ways that we see this problem. It's kind of a good mental check to see if you have understanding here. So the first thing is if we have 100% of children with blood type AB, then they must be getting an A from the blood type A parent and a B from the blood type B parent. So both of those parents must be, I guess what we could say like homozygous. They would have 2 A's for one and then 2 B's for the other, meaning 100% of their time their child would be getting an A and a B in order to be AB blood type. Okay, remember that's not always the case because these A and B parents might be a blend of A and O alleles, but if 100% of their kids are AB, they're both going to be kind of like true breeding AA and BB. Okay. All right, so another example of the same problem but with a different outcome. One parent has blood type A and the other has blood type B. What is the genotypes of the parent if half the offspring are AB and half the offspring are A? So without even having to draw a Punnett square, the first thing that should be going through your mind is that 100% of offspring are getting an A allele, right? Whether they're AB or A, they have an A allele, okay? So that should be telling you then that this blood type A parent has 2 A alleles, okay? Otherwise, we wouldn't see 100% of the offspring having at least one or having an A allele, okay? Now, on the flip side, this blood type B parent, okay, they're giving a B half the time. The other half the time, there's no B present, right? And so that tells us that they're giving us an O instead, which is why half the offspring just have blood type A as opposed to having blood type AB, like we saw in problem 2, okay? So we can then conclude that that blood type B is going to be a BO genotype, right? Because half the time they're giving the B to make the AB child, and half the time they're giving the O to make the AO child, whereas the other parent is solely giving an A 100% of the time, making them AA, okay? Hopefully that makes sense. And again, apologies. Wait, did that? Oh, I did that. Okay, I was going to say, that slide froze again. I'm going to lose my mind. Okay, good. All right. Last version of that question. There's many different ways that these A and B parents can produce offspring. So in this question, one parent has blood type A, the other has blood type B again, okay? But now we have a quarter of the offspring have AB blood, a quarter of the offspring are A quarter of the offspring are B, and a quarter of the offspring are type O, okay? So really the only thing that you need to focus on is the fact that a quarter of their offspring are type O. That tells you all you need to know. So remember, O is recessive, okay? So in order to have blood type O, you have to have 2 O alleles. That means each parent must have an O allele. So each parent must be heterozygous, if you will, or have an A allele and an O allele, and a B allele and an O allele. So if we look back at this chart, they would be AO and BO, because that's the only way that an A and B parent could make an blood type O offspring with those 2 O alleles. Okay, last question, and then we'll move on to sex-linked traits and then pedigrees. So in this example, we're looking at the blood types of Doe family. Mr. and Mrs. Doe both have blood type A, and then the Jones family, where Mrs. Jones has type A and Mr. Jones has type AB. Then we have these 2 babies. They got lost in translation at the hospital. Baby 1 has type O blood, and baby 2 has type B blood. Okay, so right away, hopefully you've kind of started to figure this question out. It'll give you about 30 seconds to read through it to try and determine which baby belongs to each family, and then we'll review. Okay, hopefully you were able to see pretty easily that baby 2 had to come from the Jones family. They have type B blood, so have to have at least 1 B allele, okay? And only Mr. Jones of all 4 parents carries that, okay? So that must be their baby. They would also be capable of making a type O baby because Mr. Jones can only give an A allele or a B allele. On the flip side, the Does could give O alleles to make a type O baby. It would just have to mean that each of these type A's is actually an AO blood type, carrying that recessive O allele, so that that baby must belong to them. It would be impossible for it to be made from the Joneses, and it would be impossible for baby 2 to be made from 2 blood type A parents. Okay, so baby 1 is a Doe baby, baby 2 is a Jones baby. Okay. Enough of multiple alleles. No questions there? Okay, perfect. So next slide then, we'll talk briefly about sex-linked traits. So sex-linked traits are also pretty straightforward, but these are going to be traits or disorders that are carried on the X or Y chromosome. Much more commonly, they're carried on the X chromosome. The Y chromosome is very, very small, and it carries very few genes, so it's much less likely that a disorder or a trait is carried on there, and much more common that it's carried on the X chromosome. Because of this, color or sex-linked disorders are much more frequent in males than females. Why is that? Well, males have an X and a Y, and females have 2 X's. So most sex-linked traits are recessive, okay? But it doesn't matter if a trait is recessive if you only have one chromosome that carries that allele. So basically what that means is because males only have that one version of the X chromosome, if they get the bad copy, if they get the copy with the disorder on it, they have the disorder. Whereas for females, because we have two versions of the X chromosome, if they get one bad copy, they will not have the disorder. They'll just be a carrier of it because they're protected by the other dominant allele. So you can see in this small example, we have a father without hemophilia. So hemophilia is a bleeding disorder, can't properly clot your blood. The father does not have it, but the mom is a carrier of it. She does not know, most likely, that she's a carrier unless she had genetic testing done. But when they have offspring, there's half of their children are going to be completely unaffected, one daughter and one son. Okay, you'll notice that one daughter got that bad X chromosome from her mom, and so therefore she's a carrier. And then this son, half of their sons are predicted to have hemophilia because they get the bad X from mom. Okay, remember that sons get the Y chromosome from dad, right? Which means that their X comes from mom. So there's a 50-50 chance that they're going to get the bad copy versus the good copy for a sex-linked trait. Whereas daughters, they get one chromosome from dad, or one X from dad, one from mom. So it's much less likely that they're going to have that bad copy. And if they do, they're just considered a carrier. They wouldn't actually show symptoms of that disorder. Okay. So really common sex-linked disorders: colorblindness, hemophilia, and Duchenne's muscular dystrophy, which is a very quickly progressing form of muscular dystrophy. All of those are carried on the X chromosome. So you can pull your classes, I always do, to ask if anyone's colorblind. Usually I have at least one male in each class who is colorblind, at least in some part. I haven't had a female, but it's not impossible, but it's just much less likely. And this is going to come into play when we start looking at pedigree trends in a few slides. Okay, so in one couple, this is an example problem. In one couple, the mother is a carrier for colorblindness. Okay, so she has one good X and one that's a carrier for this colorblindness trait. And the father has normal color vision. Okay, I wonder if I can like write backwards right in a mirror and hold something up for you guys. That's the problem. Otherwise, I would always just like write things out. See what I can do. Okay. Oh boy, this is going to be tough. Okay, so the mother's a carrier for colorblindness. So we'll have her. Can you even see that? No. Okay, never mind. We'll just talk through it. We'll figure this out for next week. So the mom has half of her X's are good, half are not. The father is normal. So what's the probability that their son will be colorblind? Okay, so in thinking about this, remember that the dad does not really matter as far as whether or not the son's going to be colorblind, because the dad's just giving his Y to his sons, which is why they're sons and not daughters. Okay, so the sons just get a Y from dad. Okay, other than that, the X's are going to come from mom. So half the time, the X that they would get from mom is going to be that normal X. Okay, because remember she's a carrier, so she has one good one, one bad one. So half the time we'll have a normal X. The other half of the time we'll have that that colorblindness gene. Okay, so we should have 50% of sons that are colorblind and 50% that have normal vision. Okay, given the fact that half of the X's from mom will be normal and half of the X's from mom will not. And remember that dad is not affecting his son at all here, because the only thing that he's giving his son is the Y. Okay, so 50% of sons will be colorblind. For daughters, here, so they're getting a good X from dad, right? Because again, if dad is not colorblind, okay, and there's a daughter, so gender is really determined by the male, so they have at least one good X. So we already know 0% of daughters then will be colorblind because they will each have at least one dominant X from dad, okay, who has normal colored vision. Okay, then beyond that, there's a 50% chance that they'll get another good X from mom. And there's a 50% chance that they'll get the bad X from mom. But even so, they're not going to be colorblind. They would just be a carrier and potentially pass it on to their future sons or daughters. Okay, so within the daughter population, 0% are going to be colorblind, 50% would be carriers. And again, you could hopefully write this out for yourself while I struggle through technology in order to see that. Okay, so that's sex-linked traits. Another example, in a different couple, the father has hemophilia and the mother has two alleles for normal blood clotting. Okay, so the father has one X right? That is the recessive hemophilia gene and one Y. And then the mom has 2 good X's. Okay, what is the probability that their son will be a hemophiliac or their daughter? Okay, so here's the thing. Remember that the father's X is never going to affect their son, right? Because he's not giving an X to the son, he's giving his Y. So the good news here is that sons are getting that Y from dad, and then the mom has 2 normal X's. So they're getting a normal X 100% of the time. So 0% of these sons will be hemophiliacs because they're not getting the X from dad, they're getting the Y from dad, and they're getting a normal X from mom, okay? On the flip side, 100% of the daughters will be carriers. Because mom has 2 good copies of X, each daughter will get 1 good copy of X, but they're also 100% of the time going to get a bad copy of X from dad, because that's all he has to give them for his daughters. So they'll be 100% carriers, but 0% of their children as a whole will be hemophiliacs. Okay. All right, so we're going to talk a little bit more broadly now. We're going to review a tiny bit about just regular dominant and recessive traits, and then we'll look a little bit more into X-linked traits, Y-linked traits, and mitochondrial traits and their inheritance patterns. So I have 5 different pedigrees up here. They're all labeled. We're going to go into each one in detail. But on the AP exam, you might be faced with just a pedigree with no label, and you'd have to determine its inheritance pattern using some clues. Okay, so those clues that you can use are what I'm going to go over with you next. Okay. So the first one is going to be for a pedigree that shows an autosomal dominant trait. If you're not familiar with pedigrees, circles are females, squares are males. Ones that are filled in are affected with whatever trait. It doesn't always have to be diseases. Sometimes it's like having attached earlobes or having a widow's peak or something like that. Okay, but individuals that possess that trait are going to be shaded in. Sometimes carriers will be half shaded, but oftentimes they won't be, because oftentimes pedigrees are made off of interviewing family members, and you don't know. Oftentimes if you're a carrier, like, let's see, widow's peaks are dominant. I have a widow's peak. I don't know if I'm homozygous dominant or heterozygous for that, so I couldn't tell you that unless you took a look at all of my offspring, of which I have none, but if I had them, you could look at them and determine kind of if I would be heterozygous or homozygous dominant. So anyway, because of that, they're usually not shown. So these are also generations from oldest to youngest. Okay, lines in between a circle and a square would be a mating pair. Okay, and then the lines coming down off of them would be their children. So this couple in line 1 had 1, 2, 3, 4, 5 children. This child, the third child, married and had 1, 2, 3, 4 children. And then 2 of their children got married and had 4 and 3 children of their own. So this is what we can tell from a pedigree. When we are looking at a pedigree, if we want to determine inheritance patterns, there are some trends that we can look for. Okay, so if a trait is autosomal dominant, and first I'll define autosomal. Autosomes are all of our chromosomes that we all share. So chromosomes 1 through 22, basically everything except for the X and Y chromosome. So unless it's a sex-linked trait, it's autosomal. Okay, so almost everything that we talk about is autosomal. So if this trait is a dominant inheritance pattern, then we would be able to tell that with a couple things. 1, it has to show up in every generation, right? And here's why. So here, our unaffected individuals have to be homozygous recessive, because the affected individuals have at least 1 dominant allele. They're either heterozygous or homozygous dominant. So if they have at least 1 dominant allele, okay, then that means they're going to pass that on, and we would see that in the next generation if they were going to pass that on to their offspring. Okay, it would be impossible for a child to get this trait or disorder, depending on what it is, if one of their parents wasn't affected, because they would have to have a parent with that dominant allele in order to have the dominant allele themselves. Okay, so it shows up in every single generation, as we can see here. Okay. If a child is affected, they must have at least 1 parent who is affected. Okay, so we'll look here, 1 parent, here, 1 parent, here, 1 parent, etc. Okay, again, because if they have a dominant allele, one of their parents must have given it to them if this is a dominant trait. And then the last thing that you can check for is that the number of females is approximately equal to the number of males. So here we have 1, 2, 3, 4, 5 females and 1, 2, 3, 4 males. approximately equal. Basically what we're looking for there is this a sex-linked trait? Because these numbers are relatively the same, we can determine that this is autosomal. Because it doesn't skip generations, we can determine that it's dominant. On the flip side, for something to be autosomal recessive, it can skip generations. That doesn't mean it always does, but it can. Okay? And so we can have affected children where both parents may not be affected. We can see that here. This child is affected with this disorder, but neither of their parents are. In a recessive trait, this is possible, right? What that means is each of these parents are carriers or heterozygotes. Okay, so when they reproduce, this offspring got both recessive copies, which is why they show the trait, even though both of their parents were protected by at least or by one dominant allele. Okay. This again does not work if the trait is dominant, because if they have a dominant allele, one of their parents must have had it. But because this is recessive, we can see that skipping of generations, like this whole second generation, does not have whatever trait or disorder this is. And if a child is affected again, their parents might not be. The reason we can tell this is autosomal is because the number of females affected is approximately equal to the number of males affected. We have one female and one male. Right, next, X-linked. X-linked may or may not skip generations. It depends. It's recessive usually for the most part, so it's possible that it would skip a generation, and we do see that here. Okay, skipping this generation. And then we do see that here. Okay, skipping a few generations and then showing up again. Okay, but here the easiest way to tell that something is X-linked is that you'll just see many more males affected than females. Okay, so in this example, we have no females affected. We have 1, 2, 3, 4, 5, 6, 7 males affected. Okay. Another helpful hint, and again, this is very rare because it's very rare that females have X-linked disorders, but if a mom is affected, all of her boys will be, because she'll be giving that affected X to all of them. And it's the only X that they get, so they would all be affected. So those are just some trends to look for an X-linked disorder. And again, sometimes they just show you the periodic table. So you need to be able to go through these bullet points in your brain and figure out what possibly could be the outcome here. All right, Y-linked is very, very rare, but I figured I would bring it up. Y-linked disorders, again, are carried on the Y chromosome, so only men are affected. It's truly impossible for a woman to be affected with this because they don't have Y chromosomes. And affected dads will have affected sons. So you'll see here, we have an affected dad, and their 2 sons are both affected. Remember, this is not a son. There's not a vertical line here. This is a husband of offspring number 6. Okay, this dad has 2 affected sons. This dad has 1 affected son. But remember, because they give their Y to their sons, if their Y is affected, all of their sons will be affected, and you'll see this kind of unique inheritance pattern. Last weird one, mitochondrial disorders. So our mitochondria comes 100% from our mom. So instead of having this blend of both parents, inside of each of our mitochondria, and remember that they have circular DNA, which is part of endosymbiont theory and evolution, which we'll review in a month or two when we get into review mode. But because we get, they do have DNA, that mitochondrial DNA comes 100% from the mom. It's in the egg when the egg is fertilized. Okay, so in this case, because you just get your mitochondria from your mom, if there's a mutation or something going on inside of mitochondrial DNA, then that affected mom will have all affected daughters and sons, all affected offspring. Whereas affected dads will have 0 affected offspring, right? Like you can see over here, this dad is affected because his mom was, and therefore all 4 children were. This dad was affected and none of his children were affected because they all got their mitochondria from mom. On the flip side, his sibling who had children, she has all affected children because her children got the mitochondria from her. Okay, so that's just another kind of interesting, unique inheritance pattern that can show up. But again, it's really important to know these patterns and what to look for and be able to use process of elimination, because sometimes you will have to do that. Not more often than like, I hardly ever see like a Punnett square problem, but I've seen a good amount of pedigree problems that still apply all of those different inheritance patterns that we talked about today, last week, and 2 weeks ago, but they just show it visually using these guides. And so these checklists will help you remember all of that information and be successful there. So I really apologize for all the technical difficulties. I promise that won't happen again. Usually I can just use Google Slides, which are way, way easier. But because I wanted to write things, that's why I use that regular PowerPoint. Next week, we're going to talk about bacteria and viruses. My major in college was microbiology, so bacteria and viruses are probably my favorite thing ever to talk about. They're really, really cool, and as much as you think you know, you probably don't know everything about them. So this is going to kick us off into kind of like a new unit where we talk about the human body, biotechnology, the immune system, nervous system, etc. So please join in. You can actually scroll down on the watch page and RSVP for next week. And yeah, I hope to see you there. Again, remember that you can sign up just for $20 to watch replays and have lots of access to my slides and all sorts of other helpful things to help you study and get prepared for your AP test. Hopefully I see you next week to talk about bacteria and viruses. Otherwise, have a great week and have a good rest of your night. Bye, everyone.